Dilated cardiomyopathy represents the ultimate mechanical failure of cardiac systole. At the macroscopic level, the heart becomes a big floppy pump, driven by microscopic muscle fibers struggling to contract against circulatory system resistance. This failing state is defined by three cardinal clinical signs. First, a severely depressed ejection fraction. Ejection fraction is the percentage of total blood volume in the left ventricle that is pumped out with each heartbeat. Second, an S3 heart sound caused by blood rushing into an already overfilled chamber. And third, a hollow systolic murmur driven by mitral regurgitation. Over time, these mechanical deficits culminate in systemic congestive heart failure. Congestive heart failure is a chronic condition where the heart's diminished output causes venous pressure to rise, forcing fluid to back up into the lungs and peripheral tissues. The pathophysiology here is built on a mechanical paradox. The heart attempts to compensate for microscopic injury by physically stretching outward to pump more blood. However, this exact physical stretch ultimately destroys the muscle's mechanical leverage. The heart's built-in survival mechanism directly causes its own fatal anatomical collapse. The entire cascade of dilated cardiomyopathy begins with a drop in myocyte ATP production. ATP, or adenosine triphosphate, is the primary molecular energy currency of the cell, and its depletion results in an immediate loss of basal contractile ability. The primary etiologies of this initial cellular insult fall into three categories: ischemic damage from poor blood flow, exposure to toxins like alcohol or chemotherapeutic agents, and specific genetic mutations in the MYH7 and MYBPC3 genes. A highly specific viral pathway also triggers this cascade. The Coxsackie B virus enters the host via the gastrointestinal or respiratory tract, rapidly establishing viremia, which is the widespread presence of the virus in the bloodstream. This animated diagram details the exact mechanism of viral docking. Coxsackie B achieves cellular entry by docking directly into the CCR receptor on the surface of cardiac myocytes. The CCR receptor is a specific membrane protein that normally regulates immune responses, but is hijacked by the virus to penetrate the cell. Once inside, the infection poses a dual threat. It causes direct cytolytic injury, literally breaking the myocyte apart from the inside, while simultaneously triggering a highly destructive local immune response, mediated by infiltrating T cells and cytokines. Clinically, this combination of direct viral replication and targeted immune damage presents acutely as myocarditis. Myocarditis is an active, localized inflammation of the heart muscle. Whether triggered by a virus, a chemical toxin, or a genetic mutation, this initial localized drop in contractility forces the heart to initiate macroscopic, systemic structural changes simply to maintain cardiac output. The immediate hemodynamic consequence of myocyte injury is incomplete ejection. Because the weakened muscle cannot fully empty the chamber during systole, an increased residual volume of blood remains in the left ventricle. To accommodate this trapped volume, the left ventricle undergoes eccentric hypertrophy. The chamber walls stretch outward by adding new sarcomeres in series, elongating the muscle fibers rather than thickening them. This stretching provides an initial benefit by increasing the end-diastolic volume. End-diastolic volume is the total amount of blood present in the ventricle right before it contracts, and increasing it raises the physical preload placed on the muscle walls. This dual-panel graphic illustrates the Frank-Starling mechanism in action. By increasing preload, the heart stretches its sarcomeres into a more optimal alignment, temporarily boosting cardiac output by climbing the upward slope of the Frank-Starling curve. The heart essentially stretches to pump more. Chronic volume overload eventually pushes the left ventricle past its mechanical tipping point. The chamber stretches beyond its functional elastic limit. At the microscopic level, this chronic stretch misaligns the actin and myosin filaments inside the muscle cells. They are physically pulled too far apart to effectively grasp each other, severely limiting cross-bridge formation. The mechanical reality is absolute. Decreased cross-bridge formation dictates a profound decrease in overall force generation. The organ's compensatory attempt to maintain stroke volume through chamber dilation physically destroys the sarcomere's ability to generate tension. A temporary volume fix becomes permanent systolic failure. This microscopic loss of force translates directly to macroscopic clinical markers. The ventricle's stroke volume drops significantly, resulting in a plummeting ejection fraction. Looking at this cross-section of the left atrium and ventricle, we see the secondary anatomical failure caused by the altered geometry. As the left ventricular cavity dilates outward, it physically pulls the mitral valve annulus apart, preventing the valve leaflets from closing completely. The immediate hemodynamic consequence of this incomplete closure is severe. During systole, highly pressurized blood escapes backward through the gap and into the left atrium. This specific retrograde turbulence creates a hollow systolic murmur. A hollow systolic murmur is a continuous whooshing sound, heard via stethoscope throughout the entire contraction phase of the heartbeat. The backflow of blood dictates a strict path of retrograde hydrostatic pressure. It moves from the left ventricle, back up into the left atrium, and then retrograde directly into the pulmonary veins. The terminal event of this cascade occurs when the backed-up pressure hits the pulmonary capillary beds. The intense pressure forces fluid to leak out of the blood vessels, across the membrane, and directly into the alveoli of the lungs. The initial systolic failure of the left ventricle dictates a predictable retrograde cascade. It eventually drowns the lungs in fluid, manifesting clinically as overt congestive heart failure. Why doesn't a dilated heart simply shrink back down to normal size once the initial insult, like a viral infection, is cleared by the immune system? The answer is fibrotic remodeling, a molecular lockdown mechanism initiated when myocardial injury activates TGF-beta. TGF-beta, or transforming growth factor beta, is a signaling protein that strongly promotes the production of scar tissue. This intracellular signaling map shows the exact surface receptor mechanics. TGF-beta binds to a type 2 receptor on the cell membrane, which subsequently recruits and activates a type 1 receptor. The activated receptor phosphorylates SMAD2 and SMAD3. SMAD proteins are primary intracellular messengers that carry signals from the cell surface directly to the nucleus. Once phosphorylated, they combine with SMAD4 to form a complex. Once inside the nucleus, this complex explicitly targets and activates the genes responsible for producing collagen and fibrin. These tough, rigid proteins are heavily deposited into the extracellular matrix. The extracellular matrix is the three-dimensional structural scaffolding surrounding cells, and filling it with collagen permanently replaces elastic, functional myocardium with rigid scar tissue. This TGF-beta-driven fibrosis structurally locks the heart in its dilated, weakened geometry, guaranteeing a one-way path of worsening mechanical dysfunction. This flowchart illustrates the complete progression of dilated cardiomyopathy. It is a single, unbroken chain of strictly governed mechanical and thermodynamic events. A cellular insult drops overall contractility. The heart attempts to compensate via outward stretch and eccentric hypertrophy. That eccentric stretch directly causes actin and myosin fiber misalignment, leading to a profound drop in contractile force, severe left ventricular failure, and a plummeting ejection fraction. The dilated geometry stretches the mitral annulus open, causing valvular regurgitation, retrograde blood flow, pulmonary edema, and finally, congestive heart failure. Dilated cardiomyopathy represents the ultimate physiological tragedy. A cellular survival mechanism is pushed past its mechanical limits, permanently reinforced by fibrotic signaling, and ultimately transforms the heart's own compensation into the engine of its destruction.